thanks for being here. George Yorling, our Chief Scientific and Mission Officer, is telling me that it's difficult to hear me. I'm going to get really close so you can see my face right up close. Welcome to HDSA's research webinar series. This is Ask the Scientist Anything. Thanks for coming today. But before I introduce our speakers, I am going to just give you some brief instruction for how to ask questions today and some other stuff about our webinar. So we're going to, actually this is incorrect. We're not answering questions after the presentation today. You can send a question anytime. We're asking questions as we go. That's the whole point of this. You can click on the chat function in your toolbar to type in your question and hit send. Only HDSA and the panelists are going to be able to see your questions. And we're going to be fielding them to our speakers today. We are recording this webinar. Somebody just asked that in the chat. Thanks for that question. You're going to be able to view it at hdsa.org/researchwebinar or also on our YouTube channel. We try to get that up within a few days. So yeah, without further ado, I want to let our speakers introduce themselves, and we'll start with Dr. Ed Wild.
Hi there, I'm Ed. I am in the East End of London, where you might bump into Mary Poppins or Dick Van Dyke, chimney sweeps, cockneys, jelly deals. Yeah, so that's where I am, and I'm Huntington's disease researcher. I've been doing research for 15 years, and I co-founded HD Buzz with Jeff Carroll. I'm also a consultant neurologist, so I'm a clinician scientist. And from the 1st of October, I'll be Professor Ed Wild because I was promoted, but it hasn't kicked in yet. So I'm still Dr. Ed Wild for like 3 more weeks at UCL, University College London. Hi.
How about Dr. Jeff Carroll?
Thanks, Sarah. Hi, everybody. Like Ed said, I co-founded HD Buzz with Ed more years ago than I really like to think about. I'm a Huntington's disease researcher. I work primarily on mouse models. I'm a basic scientist. And I've worked a lot in Huntington lowering drugs with a bunch of different companies in that space, as well as doing basic research on, how HD affects the brain and body. And I'm a member of an HD family, and I carry the HD mutation. And I can't think of anything else to say right now. Hi, nice to see everybody.
Thanks, Jeff. How about Rachel Harding?
Hi, everybody. My name is Rachel. I'm from a similar neck of the woods as Ed, but I'm actually based in Toronto now. So I'm a research fellow at the Structural Genomics Consortium, which is part of the University of Toronto. And like Jeff, I'm a basic scientist. So I'm working in the wet lab doing research on Huntington's disease. And my interest is looking at what the Huntington protein looks like and how that differs between people with different forms of Huntington's disease. And I also, should also say, recently started writing for HD Buzz. So you'll see more of the articles now are by myself and Sarah, who's going to introduce herself shortly.
Thanks, Rachel. Go ahead, Sarah.
Hi, everyone. I'm Sarah. I currently live in California. I work at University of California, Irvine. And I, like Rachel and Jeff, work in the lab on the daily, and I use stem cell models to study Huntington's disease. And so we use cells from patients to model brain cells. Like Jeff, I'm from an HD family, but I was fortunate enough to test negative. And as Rachel mentioned, we recently started writing for HD Buzz, so you're probably going to see us around more on that website.
Great. George, you want to say a quick hey as well?
Oh, I just say hello to everyone, and I'm George Yorling. I'm Chief Scientific and Mission Officer of HDSA, and today I'm just here supporting my colleague Leora Fox and everyone else here. I'm excited to be able to for us to be able to provide this as a follow-up to our what I thought was a really successful session of Ask the Scientists Anything at convention this past year. So thank you for organizing this and for all our speakers for coming today.
Thanks, George. So we actually got a lot of questions through our webinars at hdsa.org email, and I'm going to kick us off with a question that has come up quite a lot, which is about Huntington lowering. So can we start off with sort of an update on what's going on with Huntington lowering? What is it, and what are the drugs that are in the progress of being tested for HD? I know Jeff had wanted to get this kicked off, so.
Oh, that's me. Sorry. You have to do super obvious segues. I'm not that smart. Okay, so let's start with like a general background on Huntington lowering for people. So for people who've been following this research for a time, you might have heard us refer to it in the past, or other people refer to this as like Huntington silencing, which is the idea of trying to kind of shut down the gene product that's altered in people who inherit the Huntington's disease mutation. We tend not to say Huntington silence anymore, because silencing implies that like we're trying to completely shut off the production of the gene, and that's almost never the case with the kind of therapies that we have access to today and that we're going to talk about from clinical trial updates. So really it's about like lowering the amount of the mutant gene product that causes the disease. So what I mean by gene product, so every human in the world, or almost every human in the world, has 2 Huntington genes, and one from mom, one from dad. And HD patients inherit one from either parent that has a mutation or a change in the sequence of the DNA. So they inherit right from the conception through birth and development and their life. They have this mutant Huntington gene. For reasons that are still kind of an important bit of basic science, we don't really understand why it sort of takes decades for this mutation to cause changes in the brain cells that read out or emerge as Huntington's disease symptoms. But we definitely know that every person with HD has a mutant HD gene, and that is invariably associated with the eventual emergence of symptoms of HD. Ed, do you want to talk about CAG sizes and the different ranges before we talk about Huntington lowering again?
Nah. We can talk about that later if people have questions about it, but I don't think it makes a big difference when it comes to lowering, right? Okay. Or does it? Am I missing something?
No, it's okay. I was just like going to break up. I was sick of hearing myself monologue. This is banter.
Well, yeah, join the club.
So the vast majority of HD patients have a mutant HD gene and a non-mutant HD gene. And we know that assuming their mutation is of a certain severity, they will have symptoms in their lifespan. Huntington lowering is the idea of, well, let me back up. So DNA encodes for proteins. So the DNA that we inherit from mom and dad is a set of instructions to make proteins. And so when a cell needs the Huntington protein to do its job, it goes into the nucleus, the middle part of the cell, searches through the DNA, finds a copy of the Huntington gene, and makes a copy of it. It makes a copy of it because we don't like to mess with our DNA very much, because if you screw up your DNA, you get cancer. And so cells make this copy of the genetic message of our Huntington genes and export it from the nucleus out into the busier parts of the cell. That messenger molecule that carries the message out to the nucleus is called messenger RNA, because it's not in the language of DNA, it's in a closely related language called RNA, and it carries the message of the Huntington gene out to the busy part of the cell, where it's made into a protein. And proteins are sort of the functional constituents of cells. They're the little machines that make cells work. So when we talk about Huntington, it's confusing because there's a Huntington gene, which lives in DNA, and none of the therapies that we're going to talk about are going to influence that. There are like next generation, someday things like CRISPR that could potentially address that. But right now, none of the therapies we're going to talk about influence the Huntington DNA. That DNA is copied into this message, which we can potentially influence through ways we'll talk to you about, and then it's eventually made into a protein. And we think that it's the Huntington protein made from the mutant HD gene that actually causes all the problems. So if there was some way to sort of block or get rid of the mutant Huntington protein in particular, or both the mutant and the wild type, we might be able to prevent HD. And certainly in animal models, that's the case. If we can block production of the mutant Huntington protein in animals that we've given a mutant HD gene to, we can fix them. So the general idea of Huntington lowering is at some point in that pipeline, DNA, RNA, and protein, to interfere in some way. And actually now there's multiple ways. I'll pass off to Ed for a brief overview of the different types of Huntington lowering experiments happening in clinics in humans right now. And then maybe we'll pass this around because there's a lot to unpack with this.
Yeah, so I think the easiest way to keep all of this stuff in your head is to think about 3 ways in which the drugs are given. So all of the Huntington lowering drugs that are currently being worked on or tested or coming online in the next few years is delivered to the human body by one of 3 ways: injection into the spine, a pill, or brain surgery. And if I had my slide deck, I'd have a syringe, a pill, and a power drill to represent those 3 things. So all of the trials that were happening until recently were the injection into the spine ones. So a syringe that's the needle is stuck into the base of the spine. And those are called ASO drugs. So those are antisense oligonucleotide drugs. You don't need to remember that. And annoyingly, that what they're made of is actually DNA. It's a single strand of synthetic DNA. And basically that's injected into the spine. We hope it reaches the brain. We think it probably does. And it enters the cells and it sticks to the RNA. So it sticks to the message. And so you've got the RNA, which the brain has produced from the gene. And then you've got the DNA, which is the drug molecule, and they stick to each other. And when that happens, the cell detects that there's this double thing that shouldn't be there floating around in the cell, and it deletes it, it gets rid of it, and so the protein doesn't get made. So that's ASOs, and the most famous of those is the drug that used to be called HTTRX, and then it went through a phase where it was called RG6042, and then now it's changed its name again to tominersen. And that's a drug that's it was developed by Ionis Pharmaceuticals and then now is being tested by Roche. That is the one that there was all the headlines about in late 2017 and is now in a phase 3 clinical trial called Generation HD1, 800 people, which astonishingly was fully recruited during the early days of the COVID epidemic. Incredible achievement. So, injection into the spine, Generation HD1 will finish in approximately 2 years, because that's how long it takes to test whether the drugs slow progression of the disease. The other program that's already in humans, testing another ASO drug, so another injection into the spine drug, is from Wave Life Sciences. They're running 2 parallel trials, Precision HD1 and Precision HD2. Those are at an earlier stage, so those trials are at the stage where the Roche program was about 3 years ago, and they're still enrolling more participants to bring up the numbers and test higher doses. So what we know from the Roche drug tominersen is that it lowers Huntington level in the spinal fluid, which is exactly what we wanted it to do. And so far the safety and tolerability seem very good and certainly have supported ongoing testing. But we don't know whether it slows progression of the disease, and we don't know that for any of these drugs. So that's the first kind, injection into the spine, ASOs, they're made from DNA. The other one that's currently already happening, and this hasn't been going long, but it's a very much a 2020 good news virus story, is that we're in the era of gene therapy. So that is the idea of using brain surgery to inject into the brain a small dose of synthetic virus that's been scooped out, and into it has been injected the instructions for making a drug similar to the load of drugs that I just described. Instead of DNA, they're made from RNA, but that doesn't particularly matter. The point is, these are viruses that turn your neurons into a factory for making a Huntington lowering drug. And the downside is you have to have brain surgery one time to get the drug into your brain. But once the drug is in there, at least theoretically, it should carry on telling the cells to make the silencing molecule forever. So one injection should last indefinitely. And we heard, I think it was June, was it April or June? Anyway, we heard not long ago that the first patient had been dosed in the Unicure AMT 130 trial. So we know that someone in the USA has had an operation and is now the first person ever to be treated with a Huntington gene therapy drug, which is a drug that permanently introduces new genetic instructions into the brain. That several more of those are planned in the next couple of years from now my mind's gone completely blank and I can't remember the names of any of the people. Chime in, guys, with the names of some other people who are
Voyager.
Thank you, Voyager, and there's another one. It'll come to me. Spark. Good. Yay, teamwork. The third one is a pill. So basically, what we call a small molecule. I mean, all molecules are small, but small molecules are really small. No, like a small molecule would be something like aspirin, rather than like a protein, which is big. In any event, the idea is that if we can avoid brain surgery or injections into the spine, that would be ideal. It's harder to do, but it's theoretically possible to take a pill. It gets into the blood. From the blood, it gets into the brain, and once it's in the brain, it could still be so targeted that it could lower Huntington without monkeying around with too many other proteins. And a company called PTC Therapeutics has been planning this sort of approach for a while, and we're told that they are close to launching. And we hear rumors from time to time of other companies whose names may not be unfamiliar that are planning trials of small molecule Huntington lowering drugs. But I can never quite remember what's public about that, so I shall hand over to my colleagues. But those are the three things.
Roby's just chimed in. Hey, Roby, to remind us that there is a presentation on the thing that you're talking about, the small molecule drugs to lower Huntington, this Friday, as part of what's normally an in-person meeting of the European Huntington Disease Network, which has turned into, of course, a virtual event on Friday. And there's a link in the chat window for folks, and there's going to be an update about these oral Huntington lowering drugs, which will be an exciting, I think, one of the first, if not the first, public presentations about these approaches.
Yep, I can post that link.
So Rachel's a world expert in Huntington, the protein. So I feel like we should ask her one of the questions that we asked online. Not to steal Leora's chairing thing, but we're just talking about Huntington lowering. We might as well talk about mutant versus wild type Huntington. And like, is there such a thing as too much Huntington lowering?
Yeah, so a lot of the time when we're thinking about a strategy for Huntington lowering, the ideal situation would be that we only want to lower the mutated version of the Huntington protein. But the problem is that trying to differentiate between these two different versions is quite difficult to do. So in the two copies of the Huntington gene that we all have, if one of them is the mutated and one of them is just the unmutated version, there's actually the overall change between those two genes is quite small, in the same way that the overall change in the two proteins is quite small. And I mean, this is the research question that intrigues me greatly, is trying to understand how this relatively small change leads to all of these effects that we see in patients. Some of the trials at the moment, so in particular the Roche trial with tominersen, is going to be across the board Huntington lowering. It's not specific to lower any particular flavor of Huntington. It's going to be lowering all different types of the Huntington protein. Whereas other trials are going to be working on a more specific mode of action. So for example, WAVE is aiming to lower just the mutated version of the Huntington protein. So you might think that, WAVE, we currently just don't really have enough data about which one is going to be the best one to work. But what's good is that we've seen from the Roche trial data that it's safe, and we've got really good safety data of using these sort of medical interventions in patients, and patients are doing very well after being treated. So it seems like it's tolerated pretty well. But it's really, what's the best thing is that now we've got two companies who are both working with this technology, and then whichever one works, we have that available to patients. So it's kind of one of the best sort of scenarios to have. I don't know if anyone wants to. Sarah, you've unmuted.
Yes, one thing I might add is about the WAVE trials, that they target certain areas of DNA that maybe everyone doesn't have, but they have two different drugs that they're using to increase the amount of people that they can target. and someone can correct me if I'm wrong, but I think it's between like 60 and 70% of HD patients will be able to be targeted by at least one of those drugs. So they might not work for everybody, but it's a different approach that will tell us a lot. And of course, if it works, people will work really hard to develop other therapeutics that will target a larger portion of the population.
Yeah, Sarah, just
Absolutely.
to add to that, Wave just did a public-facing science update, a research update rather, which is a bit technical, but I'll put a link in the chat window for folks who want to see it, because they announced a little bit of details about their third trial to have yet another sort of chunk of the percentage that you mentioned that weren't covered to be potentially accessible, which is an exciting advance. Also, some new chemistry of their ASOs, which look quite interesting. So I'll post a link for folks to look at.
So I think one of the things that makes people a little bit nervous about Huntington lowering, so I see a question in the chat from, I think her name is Sima, sorry if I've got that wrong, but it says, could lowering the non-mutant Huntington protein have an adverse effect? And I think a lot of this sort of caution stems from the fact that in a lab situation, if we remove both copies of the Huntington gene so that cells and different systems no longer have Huntington protein, then that system doesn't survive. But that's at a really extreme version. That's getting rid of everything. And these Huntington lowering drugs are actually just reducing the amount of Huntington to a level that we hope is both safe and also should make people better as well. So it's a very different situation, and it's not like what is being done in the lab.
So one related question, and I'm going to lean way in so you can all hear me properly. A related question
We should all
about all of these Huntington trials is about compassionate use. We get lots of questions at HDSA about whether it will be possible for folks to take these drugs before the trials are complete, and under what circumstances. So perhaps I know Ed has a lot of experience speaking with families about this, and so maybe you want to start that off, Ed?
Yeah, my experience is, I mean, my medical license is European, English. We are still part of Europe, sort of geographically, if nothing else. Anyway, so the US system is rather different, and I know that there are slightly different rules around compassionate use or the right to try stuff. The bottom line is that the entire focus of the companies that make these drugs is on making enough drug for the trials. And for Roche, for instance, who are running the Generation HD1 trial, weren't really in the ASO business until they started testing this drug and making a supply chain that's capable of manufacturing enough ASO to the exceptionally high standards required to be given to humans by injection into the spinal fluid is a huge exercise. And they've had to go from 0 to 800 patients being treated every 2 or 4 months. Plus they have to make a supply of placebo drug, which is a powder that looks like the drug but isn't the drug. So the entire focus of the basically there aren't any companies that I'm aware of that have any compassionate use programs, and so far that's largely been because the entire resources of the company have been focused on designing, setting up and running the trials. The next focus for all of these, to answer a question that came up in the chat, is going to be expanding the trials to try and prevent onset, so to treat people before symptoms. And of course, if a drug gets licensed, then the companies will have to finish the trials and potentially keep running the prevention trials, while also then having enough supply chain to meet the demands of the people to whom the drug's actually being prescribed once it's licensed by the FDA, etc. So the bottom line is, I get that, look, if I had the HD mutation, I'd probably want to be injected with these drugs myself. But we don't yet know whether that is, the fact is, we don't yet know whether that would be a compassionate thing to do. And there may well be people in whom injecting these drugs for various reasons that are to do with individual genetic differences between people or other conditions that people have or doses or whatever. There may be reasons why injecting these drugs could actually be harmful. We are still in the position where we genuinely don't know whether a drug is helpful or harmful or does simply nothing. And that's true of all of these drugs. So compassionate use, as I understand it, really comes in when we have a strong suggestion that a drug is likely to be clinically helpful overall, but hasn't yet been fully tested and qualified in a particular population. I don't think we're at that point yet. It may come, and as I understand it, that that sometimes comes between an initial clinical trial result and the licensing of the drug, or the rolling out of the drug becoming available for widespread prescribing. Guys, help me out here on the American specifics of compassionate use / licensing.
I think I would just say that, like, I think you're right that what it would take is a pretty significant amount of evidence that the drug is effective before it would even be worth trying to pursue anything. And I think, based on the track record of these companies, there's going to be a fairly small window between anyone knowing that, like even the company who's running the trials and the physicians and the participants, and all of us knowing that, and the approval of a drug if it actually had a benefit. So I think it's not something I, as a HD mutation carrier, focus on a ton, because I think, if these drugs look great, they're going to get approved. There's a track record of ASO drugs getting approved for brain diseases by the FDA now. There's a clear path. All of these drug companies are very smart about regulatory approach. Like they've got very smart people working on this, and there will be as short a time as possible between, definitive proof that they work and me and other people being able to access them. And so I don't, I'm not super stressed about the couple weeks in between or the couple months. I'm sort of focused on the long term in the whole community, and really just what we have to do is get these trials done and prove that the drugs work. And as you said, the Signal trial, 800 people and in lots of people, so we know for sure they work.
Generation trial, not Signal. Signal is a different trial.
Sorry, someone asked about Signal trial, and someone asked, sorry, segue, because I wanted to address that question from Virginia about the Signal trial. Virginia asked if we knew anything about the updates of the trial, which she said that she had heard recently that the trial had finished. In general, no one except for the company and the people involved know anything. There's really good regulatory and financial and legal reasons why they have to be very, very quiet. And so as much as I would like to have an inside track, more or less, unless we're one of the professionals running the trial, we hear roughly the same time families do. So we don't have any news. And as soon as we do, certainly for trial announcements, we'll do a story on Buzz right away.
Wasn't there an announcement that the
HSG upcoming HSG study group meeting in October, I believe it is, they're on the agenda to present the results of the Signal study. So it should be this fall, late fall.
We'd heard it from previous occasions, a suggestion of slowing of brain atrophy. In other words, so atrophy is the rate of shrinkage of the brain, and so Signal is an antibody drug. It's not targeting Huntington directly, but it's targeting a receptor on the cells in the brain. And speaking purely for myself, I must say it's very interesting whenever we hear about a drug that can influence the rate of brain shrinkage, right? Because you would automatically think Huntington's disease is a neurodegenerative disease. The brain is therefore shrinking more quickly than it should. If you give the drug and the brain shrinks less quickly, that sounds good. And I think that's an understandable reaction. But there are other things a drug can do to the brain that might give that effect without necessarily actually slowing the progression of the disease. For instance, if you scan my brain now, and then I take a drug, and then you scan it in a year, and my brain is bigger a year from now, two possible interpretations. Number one is the drug has made my brain grow because it's made me cleverer. Number two is the drug has made my brain swell because it's activated the immune system. And we know that that can easily cause a sort of artifactual signal. So people's brains are not growing in this Signal trial, but we're hearing that there may be a slowing of shrinkage, and inflammation in the brain could easily do that. And we do know that these antibody drugs, if they get into the brain, can be prone to causing inflammation. So what I would say is enter the HSG convention with an open mind, but bear in mind that these indirect signals like brain atrophy, or indeed like the mutant Huntington concentration spinal fluid, there are always ways that life can throw confusing curveballs in our direction. I used an American sporting analogy, guys. Are you proud of me? Thank you. That's from American tennis, I believe.
Thanks, everyone, for thoroughly addressing that. I'm wondering if we could switch gears a little bit, and I'm thinking that possibly Sarah might be a good person to talk a little bit about this question related to stem cells. Jeanette and also some folks who wrote in have asked about where those stem cells come from. And I see you all leaning in, so I'm going to lean in closer so you can hear me once again. I'm sorry about that, everyone. So maybe you could talk a little bit about when you're doing stem cell research, where do those cells come from? Are they coming from embryos? Are they coming from adults? And what are the rules around that? We often get people asking whether they can donate embryos to research. So I'm also curious if you have any insight into that.
Sure. So when stem cell research first came out, we were getting stem cells from embryos, but I think those stem cell lines are still around, but we don't typically use those anymore. And where those stem cells come from is, for example, if you have HD and you want to have a healthy baby and you go through in vitro fertilization and you get pre-implantation genetic diagnosis, you then move forward with implanting babies that don't have the mutation. You might also have extra embryos that do have a mutation. And so when people come to that decision, they're presented with a choice. They can either keep the babies for whatever reason, the embryos, or they can donate them to science. By and large, most people choose to donate them to science. And so when stem cell research first started, before even I was in grad school, so I can't give you an exact year, people were using those lines. And so we were kind of not stuck with, but we were only offered lines that had the number of CAG repeats that happened to have been in those embryos. So fast forward to about 2006, 2007, there was a researcher named Jamie Thompson here in the US, as well as Shin Yamanaka, who was in Japan, and they discovered a way to turn skin cells back into stem cells. And so that really revolutionized the way that HD scientists can study stem cells, because we take skin cells from patients. So we used to take a biopsy from the hand, but because that gets a lot of UV radiation, we now take one from underneath the armpit. So it's just a punch biopsy. It gets chopped up into tiny little pieces, and then certain biological factors are added to those cells to kind of force the biological timeline backwards. And so you can think about all of this as like a hill with a ball at the top. And so your stem cell is the ball at the top, and it really has the energy to roll down the hill and kind of land in different crevices that are different cells. So for example, if you have a ball at the top and it rolls down and lands as a skin cell, those biological factors you add can push the ball back up the hill. So then you can make it go down a neuronal lineage. So you can make it become a brain cell. And so today, all of the stem cell lines that I use in the lab come from patient skin cells. And because patients have a variety of different CAG repeat lengths, we can then study a variety of CAG lengths. But again, that limits us to whomever walks in the door, whatever their CAG length is. So even more recently, there's been technology that allows us to modify how many CAG lengths there are. And so you guys probably know people are all different, right? Like we might have different length hair, different color eyes, and there's small variations in our DNA that lead to that. And so when we study cell lines from different people, we're also studying those small differences. So that newer technology that allows us to just modify the CAG length allows us to study stem cell lines that are completely identical except for the CAG length. And that, again, really has revolutionized stem cell biology for Huntington's disease, because with those, we'll know whatever difference we see is conclusively from the CAG mutation, the HD mutation in the CAG length. So that's where we get our cell lines. And I saw somebody in the chat wrote, like, how much does it cost to do this stuff? Nothing. It costs you nothing. We want your cells. We want people to come give us their cells. We get all of our cells from Cedars-Sinai in LA. There's a lab there that reprograms them for us, and they have a repository. So the HD field, as you guys probably have learned by now, is super collaborative. Everyone is super amazing. And so everyone that studies stem cells in the HD field started this consortium, and they basically work together and they give each other cell lines. And so that bank is kept at Cedars-Sinai, and all of the reprogramming is done there, and they kind of dole them out. So you can go online and purchase them with grant money, and they'll mail you, not anyone, but researchers, and they'll mail you a vial, and you can then do the experiments for that. So that's where we get the cells.
Sarah, what about stem cells as a treatment? Is that something that you're working on, or can you talk about how we get from stem cells as a research tool to stem cells as a treatment?
Sure, yeah. So everything I just mentioned was stem cells as a tool, but there are people that are interested, and I work with Dr. Leslie Thompson at UC Irvine, and she happens to have a preclinical trial going on for stem cell transplants, but that's preclinical, so it's just stuff in mice right now, but it's really promising. And so basically the idea is that obviously in HD you have a loss of cells, and stem cells offer not only the replacement of cells, but they can offer the replacement of factors that keep other cells that are still in the brain healthy. And so the idea is kind of twofold. You're resupplying cells to the brain, but you're also supplying cells that secrete things to keep the brain healthier, to maintain the cells that are there. And so there has been some history. I do want to differentiate between something. So there's been some history of cell transplantations in HD that traditionally a long time ago, I think even in like early 2000s, was done with fetal tissue, human fetal tissue. That's very, very different than stem cell transplantation. That you're taking from something that's not of your own body. And so you would have to give whomever you're putting that into immunosuppressants. It's just like an organ transplant. It's something foreign in your body. Stem cell transplants have a way to revolutionize that, because you can take a skin cell from that patient and then you could then put it in their own body. The caveat there being, obviously, if you're taking skin cells from an HD patient, the new cells also have Huntington's. So you would have to use that in combination maybe with some other therapeutic like Huntington lowering or even CRISPR to modify that. So yeah, so stem cells really have kind of two avenues. They're traditionally used as a tool, but there's a lot of research coming out showing that maybe there'll be some application for therapeutics. And if I could quickly, because I'm in California, give a plug for CIRM, which is the California Institute for Regenerative Medicine. It's a California-based initiative that has done a lot to fund stem cell research in California, which is why a lot of people in California study stem cells. But it went on the ballot in 2007. It was a $3 billion initiative. And Leslie Thompson, whose lab I work in, she's done really well getting grants from CIRM. And that's really helped further her research in this arena. So she recently got a $6 million grant to keep this going. They've done a lot to fund stem cell research for HD. And any Californians that might be listening, CIRM is having 2.0 come up on the ballot in November, and it will provide, I think it's like between $5 or $7 billion for stem cell research. So definitely vote for it, because they're doing a lot for stem cell research in HD.
What people should vote on what? If they're in California.
I think it's Proposition 14, but you'll have to read like your local ballot, but it is for CIRM, and it's to fund stem cell research in California. Specific audience, cool. But hopefully that affects someone.
Every vote counts.
I think we got some folks tuning in from California today. Thanks, Sarah. We actually got a couple of different questions about, this is again about wild type versus mutant Huntington. So maybe it's a good question for Rachel if you happen to know about this. A couple of people have asked if it's possible to increase the amount of good Huntington at the same time that you're lowering the amount of bad Huntington.
Yes. Well, so often when we're doing drug discovery and thinking of sort of tractable ways in which to design therapeutics, the easy route that science takes is to get rid of the bad thing or stop the bad thing from working too much. It is much more difficult to design a therapeutic or some kind of medicine that will make the good thing work more or make more of the good thing. And that doesn't mean that it's impossible, but it means that it's just much, much, much more challenging. There are ways that people do this kind of work in other fields. So for example, in the field of epigenetics, there are ways that you can get certain genes to be switched on more by changing the way that they're bound up in our chromosomes, or if the gene is more open and exposed, then you can make more of that gene. As far as I'm aware, I don't know of any research that has shown specificity of showing more of the unmodified Huntington being expressed in any system. That's not something I know of. It doesn't mean that it's impossible. It just means that that would be a very challenging project to do. So yeah, that was something that I'm sure, like, in the future, people will be looking for these kind of things and screening these kind of things, but it's not something that I know of in the preclinical pipeline just yet.
It's worth saying also, even if you could do it, we still don't know whether we actually need to. Yeah. So at the moment, the two things are either, if this is good Huntington, this is bad Huntington, one of them's doing that, so it's lowering them both, and you end up with a lot less overall. And overall, when we've tested that in animal models, that helps. The other option is to do something like that, where you lower the bad one. Wait, anyway, let's say this is the bad one. You lower the bad one more and the good one a little bit. And so you've still reduced the Huntington overall, but you've got rid of the bad one more. And so far, either of those still has every possibility of being an effective therapy. It'll be a problem for sort of later to see whether pushing the good one back up gives you any additional benefit, I guess. At the moment, if you've got 100% of good and bad, going to 200% good, there's no signs that that would be a useful thing to do, because it's the bad guy's still around causing damage. But we could be wrong about that. We've certainly never done that in people. There was something that I thought that Jeff would want to say about that. Good Huntington.
I was going to ask you, Ed, because we know from the Roche clinical trial data that the levels of Huntington that we see in different people can also be quite variable. Yeah. So does that give us an idea of whether more of different types of Huntington is better or worse? Because you see these big changes, right, in the amount that you have between different patients.
Absolutely. Not only that, but so some people seem to have lots more Huntington than other people, but also within the life of a Huntington mutation carrier, the level can change, and actually the level seems to increase as the disease goes on. And partly that may be that there's more Huntington being released from brain cells into the spinal fluid, but it could also be that the genetic instructions that make up the Huntington, that tell the brain how to make Huntington, are actually changing during the life of that person, which can actually mess up the signal that we get from the technique we use to measure the concentration. So that stuff is really complicated. But what we do know is that the more Huntington you seem to have in your spinal fluid, the more likely it is that you'll have bad symptoms at the time of the measurement. What's less clear is whether the level predicts how quickly you'll progress. And that's something that my group is working on. And it's not a great predictor of progression, but it does seem to be able to tell you to some extent what may happen. Certainly what all of this points towards is that lowering Huntington is good, even if it means lowering both. And the other thing is, the point about all of this is that this is something we can measure in people. There's a lot we can't measure in humans, but we can measure someone's CAG repeat length in the gene, and we can measure the Huntington concentration, and we can do that as many times as we like. So I think, at the moment, everyone in the trial gets the same dose of drug, whatever their level was to start with. But I think eventually we may end up in a place where we treat Huntington's disease a bit like diabetes, and we talk about Huntington control, So if you're diabetic, you measure your sugar level every day, and then every few months you have another blood test that gives an impression of how you've been doing on average with your sugar control, glucose control. And we may end up doing something similar where we're sort of tailoring the amount or frequency of dosing with these drugs to each individual's own Huntington level, and that would be quite cool.
Thanks, everyone. There are two topics that we've got a lot of questions on, and since we're getting towards the end of our time here, I want to make sure to address them. One is about lifestyle changes, and one is about somatic instability, or genetic modifiers, perhaps. So let's start with this one. So we got quite a few questions from people about whether there's anything they can do on a day-to-day basis to change how their symptoms are appearing or might appear in the future. So maybe let's address this from a research perspective. Is there any research into anything like alcohol and drug consumption, different kinds of exercise, meditation, diet? Is there anything that's really showing that folks can do to change the course of HD right now?
Ed,
I'm supposed to assign someone to answer this. It sounds like Jeff's going to do it.
No, I was going to say, Ed, do you want to do a sort of a clinical version of this, and then I can talk about a personal one, and then maybe I can talk about the somatic instability question, Leora, if that's okay.
Yeah, I mean, in terms of the research, the first thing to say about all of this stuff is what we call environmental modifiers. Nothing to do with the ozone layer, but it's stuff in your own personal environment, your lifestyle that you can change in order to change your future. And the first thing to say about this stuff is if there is stuff out there that makes a difference, it's really, really difficult to study in humans, because like you can get 50 mice and give half of them access to classical music, and the other half they live in silence, and everything else about the mice is identical. And then at the end of that experiment, you can say, oh well, the mice that listen to music did better, so that's good. But of course, humans are all genetically different from each other. We all have different diets, different lifestyles. We take different drugs, different medications. We do different forms of exercise. Our lives are so kind of messy when you try to compare them with each other, that even the biggest studies that we have, things like the Enroll HD study, which I think has 20,000 people involved now and is studying people year after year after year, it's actually really difficult to get enough data to give us what we call signal over noise. In other words, so let's say it's classical music. Like, you can ask people what music they listen to, but you know, even if you found 5,000 people who listen to classical music and 10,000 people who don't, there still might be so much that's different about all those people. And then you've got to keep following them up for 1, 2, 3, 5, 10, 20 years. So the scale of research that you need to answer these questions robustly is huge, which is not to say it's impossible. And what I think is needed is really big data sets like Enroll HD, and really smart statistical methods of looking at these issues. And it may come down to things like using artificial intelligence algorithms and machine learning, which someone's asked a question about, to basically probe these data sets in a way that is completely unbiased and sort of uses the data sets to simulate clinical trials, dividing people, making sure they're matched, and then following them throughout the disease. Short answer is there's been a few reports saying things like alcohol consumption is linked to worse progression in Huntington's disease, or that a Mediterranean diet, I think, was one of the others that was supposedly linked to more favorable progression. The reality is that we haven't really ever had a big enough data set using robust enough statistics, to my mind, to enable us to actually have useful information to give to people. And so here we are in 2020, people are being dosed with gene therapy, and I can't tell you for sure whether smoking is bad or good for people with Huntington's disease, not because it's not obvious, but because the experiment's never been done. And so the advice that I have to give my patients as a clinician is, well, actually, from general studies in millions of human beings, we know that there are some things that are good for your brain, and those things are generally things that are good for your blood vessels. And it's really boring stuff. It's exercise, it's a balanced diet, it's don't smoke, it's if you drink alcohol, do it in moderation. It's probably don't be a professional boxer, or take part in activities that involve smashing your head against walls or boxing gloves if you have the HD mutation, because your brain cells are more vulnerable. And that's it. There's I don't know of anything that you can take that will for sure reduce your risk of delay the onset or progression of Huntington's disease. That's why we really need everyone to get on board with these clinical trials, particularly now that they are of drugs that are targeting the known core mechanisms that drive HD progression. If you want to paint a better future for yourself and for the next generation, research participation is as far as I'm aware, the only evidence-based suggestion that I can make.
Thanks, Ed. And I'll just say, again, as someone sort of in this situation of having thought a lot about what I could do to try to help myself and my siblings and the other HD folks that I know, when I first got into the HD world and started reading about research, I got really obsessed with like all these different supplements, and I was taking creatine. People have been around for a while. I remember all this stuff. And creatine is not easy to take as an example. People were drinking like 30 or 40 grams a day of creatine, which isn't a drug. It's like a milkshake, and people were having really serious like bowel and intestinal problems. And, this stuff's made in some lab in China, and it's not quality controlled, and God knows what else is in it. And then furthermore, we have, one of the largest studies ever in HD of 700 something people. And that study failed, full stop, to prevent HD or to slow it. And so we know now definitively creatine doesn't help HD, and a lot of folks put themselves at risk for taking something, based on some mouse studies and some good ideas that were solid science and worth doing. And I've gotten to the point where I don't do anything special. I aspire to do the things Ed just mentioned, but neither me nor my family do anything. There's no special supplement or special technique that anyone has. Unfortunately, until we have effective drugs, the best thing we can do is just stay as healthy as we can and stay as involved as we can. As I think Ed said, that's part of it. Being involved makes me feel a lot better. And I think that's one of the best things folks can do. And I'll just segue to say something really briefly about research participation and somatic instability. So one of the benefits of having this massive study like Enroll is that we can look at not just environmental factors, but also other genetic factors in all of the folks that are in the Enroll study. And so the most recent publication that I'm going to talk about has something like 6,000 HD patients. And I think there's another study coming with something like 9,000 HD patients. And so we know what causes HD, right? It's a mutant HD gene. But it's also clear that some families have an earlier and some have a later onset than you would expect. And it's kind of been known colloquially, like people notice that tends to run in families, which suggests that it's genetic, but no one really knew why until these giant genome-wide studies where we can analyze someone's entire genome and look for genetic variants in and outside of the HD gene, not the mutation that causes the disease, but other changes in the person's DNA that are associated with them having an earlier or later onset. And if you look at enough people, you can find even really rare variants that might make someone have onset of HD earlier or later than it would be expected. And in fact, this large study, the GEM HD consortium did this study, they published it, and they found several genes that influence HD onset, actually making it worse and better, which is interesting, as well as variations within the HD gene that we don't have time to talk about today, but that built a convincing story that besides the HD mutation itself, one of the things that seems to drive earlier onset of HD is changes in the cell's ability to repair DNA. And so a number of these genes that popped up as modifiers of the age of onset of HD were genes that tell a cell, that help a cell repair its DNA, which is of course a key part of being a healthy cell. Is you've got to maintain your DNA. And based on this study and the really convincing data, I know multiple companies have launched, and they're obviously earlier stage than say the Huntington lowering programs, but are already launching human observational studies to try to follow this up and bring it to the clinic, because the implications of the genetic data really suggest some very obvious targets of genes that we might want to shut off or dampen down in order to mimic the sort of natural experiment that happened when people inherited those rare genetic variants that influence their DNA repair genes. And so there's a company called Triplet, another one, Locus 23, some other companies that have launched already just based on just the human data and the really, invaluable information that HD patients gave to the Enroll HD study and the GEM HD consortium that came out of it. So, there's a funny link between research participation and really the identification of new and exciting targets for HD research.
Jeff, this is quite new to all of us, really. DNA repair isn't something that many people, even in HD research, were thinking about 5 years ago. Are you saying that people with the HD mutation have like faulty DNA repair, or what's going on there? Like, what's abnormal?
Yeah.
So
that's,
help?
As far as we know, the answer is no, which is good, because then we'd have all kinds of other problems like tumors and cancer and stuff like that. So the model that's emerging, and this is new, this could be wrong, but I think a lot of scientists are thinking in this direction, that we have these little machines that kind of feel along our DNA and are feeling for damaged areas from, radiation or toxins or just a mistake of the DNA replication machinery. And so there are these little bulges and mismatches and stuff that happen in DNA, millions of times a day, and your cells just fix them. And it seems like what happens is those long repeats, like the one that cause HD, and expansion of the CAG tract, sometimes get recognized by that machinery. And it's not 100% clear how that leads to disease, but there's some suggestions that one of the things that happens is something we call somatic instability. And soma just means it happens in your body cells, not in your germ cells, your sperm or your egg cells. So in some of the cells in our body, the CAG tract gets longer as you age, and that includes the vulnerable bits of the brain that we think underlie HD symptoms. And so it's almost like the model that we're thinking is that these DNA repair processes recognize the CAG tract, and as they try to kind of fix it, they screw it up and make it longer. It makes the length of the CAG unstable, not in a way that's going to influence your kids, but in a way that influences that cell. And so that's the process, that a normally good process of DNA repair, that potentially is contributing in a bad way to the lengthening of the CAG tract, but only in certain cell types.
So it's not that people generally have good or bad DNA repair like skills.
Right.
It's that the accidental adding of extra CAGs is a thing that can happen to anyone, because actually the CAG tract in the Huntington gene naturally tends to favor that kind of expansion. But there are people who are slightly better at preventing that particular kind of mistake, and that's what these drugs are trying to enhance,
right?
I guess.
And by sheer chance, several or dozens people in the Enroll-HD study happened to have a tiny variant in one of their DNA repair genes that made it a little better or a little worse at doing that. And we were able, because we had such a huge sample, to say, oh, I noticed that when you inherit that, you also got HD a bit later. Let's try to do that, right? And so that's a really direct line from the genetic studies to the drug discovery.
Cool. Well, we're coming up on the end of our time here. So there's one question that we got that I think is this great typical interview question, which is, where do you see yourself in 10 years? Where's the field going to be in 10 years? And I wanted to just offer a little bit about that to kind of wrap things up. Oh, hello, Ed's dog.
He heard me talking.
It's always dangerous to kind of give exact timelines for any of these therapies that are in development right now. But there's a lot of hope in all of them, and there's a lot of planning that's going into being ready for the eventuality of having genetic therapies, of having therapies that slow HD. And that is an aspect of research that's not often discussed, but all of the companies involved and all of the major players involved in HD and HD research are thinking really hard about what happens when there is a therapy. Let's make sure that it is accessible. Let's make sure that we're enabling professionals to have more information about HD. Let's increase the knowledge of the community. Make sure that there are enough genetic counselors with experience in HD for the eventuality of folks wanting to get tested more. And these are things that HDSA is focusing on, lots of these companies are focusing on. So that, I think, is the best way to think about what's happening in 10 years in terms of medical stuff. If anyone would like to say anything final around that or anything else, please kick in.
I saw a couple of questions in the chat talking about ML, machine learning, and AI, so artificial intelligence. And this is something that in the field of neurobiology, there's been a huge amount of interest both by private organizations, like those being run by Elon Musk, which are morally questionable, but there's also a huge number of academic groups at various universities all over the world who are doing some really amazing work on understanding the human brain, both through the Human Brain Mapping Project and other projects where technology is basically leading us into this next generation of neurobiology and understanding of neurodegenerative diseases. And we're very fortunate to be at a very interesting time where there's this really amazing intersection between big data and these huge supercomputers who can process data sets like we just couldn't even do 2 or 3 years ago. And so I think in the next 10 years, there's a lot of hope that this might give us new things to be working on, or new things to be thinking about, or new ways for patients to be helped in the future for not just Huntington's disease but other neurodegenerative diseases as well. So yeah, I would keep an eye on the news because there's lots of really interesting things happening in that space, and it's moving forward at lightning speed.
Thanks, Rachel. With that, we're going to wrap this up. I want to thank you again, everyone, all of our panelists, for answering everyone's questions. We got so many wonderful questions. We will do our best to follow up. I know we can kind of send a mass email to everyone who attended today. We're also going to have this recorded webinar available on HDSA's website and on our YouTube channel. So with that, I'm going to say goodbye to everyone. Thanks for attending this HDSA webinar. Ask the Scientists, and we hope to see you again next time.
Bye, guys.
Bye.
See you
Bye.
soon. We'll do it again soon.
Bye.
Thank you, everyone.